In part 1, we had seen that adaptive immunity is made up of humoral immunity and cell-mediated immunity. Humoral immunity acts against extracellular antigens or pathogens. Cell-mediated immunity acts against intracellular pathogens or antigens. So when a pathogen has succeeded in entering some of our cells, this pathogen will be dealt with by cell-mediated immunity. And cell-mediated immunity is the function of T lymphocytes or T cells. The T cells possess receptors known as T cell receptors. We have got billions of different T cells, and every cell possesses a unique T cell receptor which is complementary to a particular antigen. A number of receptors may be present on the same cell, but they are identical. They will recognize only one particular antigen. So this T cell receptor, every T cell receptor has got an antigen binding site or antigen binding cleft. And the antigen which is recognized by the T cell receptor is generally a peptide formed from mostly microbial proteins. So this antigen binding site of the T cell receptor or TCR is complementary to a particular antigen, mostly a peptide. While this T cell receptor is complementary to the foreign antigen, it cannot bind the antigen if the antigen is present as a free molecule. The T cell receptor will bind the foreign antigen only when it is combined with a self protein, which is known as MHC protein. These MHC proteins are present on all the cells. They are present on the surface of all the cells, and they are unique to each individual. So when the foreign antigen is combined with a self MHC protein, this complex is recognized and bound by a particular T cell receptor which is present on some T cell. These MHC proteins are of two types, MHC1 protein and MHC2 protein. MHC1 proteins are present on the surface of every cell. These are transmembrane proteins which are made up of an alpha subunit and a beta subunit. The alpha subunit possesses the antigen binding site or the peptide binding site or cleft. So this cleft or site is constituted by two domains of the alpha subunit, which are alpha 1 domain and alpha 2 domain. MHC2 proteins are present only on the surface of B lymphocytes, macrophages, and follicular dendritic cells. These proteins are also transmembrane proteins, and two domains, alpha 1 domain and beta 1 domain of the MHC class II protein form an antigen binding or peptide binding cleft, which is complementary to a particular antigen. We have seen earlier that the T lymphocytes can be of several types: cytotoxic T cells, also known as killer T cells, helper T cells or T helper cells, suppressor T cells, and memory T cells. Each of these types performs a specific function. We will first look at the function of cytotoxic T cells. The function of cytotoxic T cells is to destroy the infected cells So if a pathogen has succeeded in entering some of our cells, these cells have become infected. The cytotoxic T cells will destroy these infected cells, and along with the cells, the pathogen which is present inside these cells will also be destroyed. So, infected cells plus the pathogen are destroyed by the cytotoxic T cells, and this prevents the spread of infection to other healthy uninfected cells. If the infected cell is not destroyed, the pathogen will multiply inside this cell. After some time, this cell will rupture. The infected cell will rupture, the pathogen will be released. And this pathogen can then infect other healthy uninfected cells. So by destroying the cells having the pathogen, the cytotoxic T cells prevent the spread of infection to other healthy uninfected cells. Most of the viruses and many of the bacteria reside in the cytosol of the infected cells, and they multiply in the cytosol of the infected cells. Their proteins, the microbial proteins, are broken down by 26S proteasome. So when the protein is broken down, a number of peptide fragments are formed. These peptides are picked up by MHC1 proteins, and they are bound firmly to the MHC1 proteins, on the peptide binding cleft or the antigen binding cleft of the MHC1 protein. And then this MHC1 protein is inserted in the cell membrane, and the peptide which is bound to this MHC protein is displayed on the surface of the infected cell. The combination of the foreign peptide plus the self MHC1 protein will be recognized by some cytotoxic T cell which has got a T cell receptor complementary to the foreign peptide. So this T cell receptor will bind to the complex of the foreign peptide and the self MHC1 protein. So we have a MHC1 protein here, which is present inside the lumen of the endoplasmic reticulum. And this is a foreign protein, which is broken down by 26S proteasome. And when it is broken down, a number of peptides are formed. Now, one peptide binds to this particular MHC1 protein, forming a complex made up of a foreign peptide and a self MHC1 protein. This complex travels to the cell membrane of the infected cell. The MHC1 protein gets inserted in the cell membrane, and this foreign peptide is displayed on the surface of the infected cell. This is the cytotoxic T cell having a T cell receptor complementary to the foreign peptide or the foreign antigen. So this T cell receptor will bind to the foreign peptide plus the self MHC1 protein. In addition to T cell receptors, the cytotoxic T cells also possess a co-receptor. This co-receptor is also a transmembrane protein, and it is known as CD8. CD is cluster of differentiation. So this CD8 protein is made up of two polypeptide chains, an alpha chain and a beta chain, which are joined to each other by a disulfide bond. And this CD8 protein, just like the T cell receptor, has got a cytoplasmic tail, a transmembrane region, and an extracellular portion. So the extracellular portion of CD8 binds to the MHC1 protein. In fact, it binds to the alpha 3 domain of the MHC1 protein. The cytoplasmic portion or cytoplasmic tail of CD8 is associated with an enzyme which is LCK. LCK is a tyrosine kinase which is normally inactive. The cytoplasmic tail of T cell receptor is associated with a complex known as CD3 complex. Now, this CD3 complex is a transmembrane protein made up of several polypeptide chains. These chains are a gamma chain, a delta chain, two epsilon chains, and two zeta chains. So together, these chains constitute the CD3 complex, which is a transmembrane protein associated with the T cell receptor. The cytoplasmic portions of gamma, delta, epsilon, and zeta chains possess immunoreceptor tyrosine-based activation motif, which we have seen earlier. This motif is made up of two tyrosine residues which are present in the polypeptide chain at a distance of 9 to 11 amino acid residues. So when this binding occurs between the T cell receptor and CD8 protein on one side and the foreign peptide plus the self MHC1 protein on the other side. Upon this binding, LCK becomes active. LCK, which is associated with the cytoplasmic tail of CD8. So this LCK now becomes active, and this active LCK phosphorylates the items of CD3 complex. And these phosphorylated items then attract and they act as a docking site for a protein known as ZAP70. ZAP70 is zeta associated protein of 70 kD. The molecular weight is 70 kD. So we have an infected cell here. This is the cell membrane of the infected cell. This is the MHC1 protein inserted in the cell membrane, and this is the foreign peptide bound to and displayed by this self MHC1 protein. We have a cytotoxic T cell. This T cell has got a T cell receptor which is complementary to this foreign peptide. So this T cell receptor binds to the foreign peptide plus the self MHC1 protein. This is the co-receptor, the CD8 transmembrane protein. So the extracellular portion of CD8 binds to the alpha 3 domain of the MHC1 protein. And this is the enzyme LCK associated with the cytosolic or cytoplasmic tail of CD8. But this T cell receptor is associated with the CD3 complex, which is made up of this epsilon chain, this delta chain, this gamma chain, this epsilon chain, and these two zeta chains. And these shaded portions, these are the items which are present in the cytoplasmic tail of the CD3 complex. So when LCK becomes active, it phosphorylates the items, and these items then attract and bind ZAP70. Now this ZAP70 becomes active, and this ZAP70 is also a tyrosine kinase. So this active ZAP70 now phosphorylates the tyrosine residues of some target proteins which are present in the cytotoxic T cell. And this phosphorylation results in the release of some granules which are stored inside the cytotoxic T cell. So these stored granules are now released from the cytotoxic T cell, and they are targeted at the infected cell. So these granules will act on the infected cell. The granules which are released by the cytotoxic T cell contain three proteins: perforin, granzyme, and granulysin. So several molecules of perforin, several molecules of granzyme, and several molecules of granulysin are released from the cytotoxic T cell, and they are directed at the infected cell. Now, several molecules of perforin get inserted in the cell membrane of the infected cell, and they are polymerized to form transmembrane pores, tubular structures inserted in the cell membrane of the infected cell, which act as transmembrane pores. So the contents of the infected cell leak out through these pores, and granzyme and granulysin molecules enter the infected cell through these pores. So this is the polymerized perforin or polyperforin, which is inserted in the cell membrane of the infected cell and which has formed a transmembrane pore. So the contents of the cell leak out through this pore. Granzyme and granulysin enter the cell through this pore. This granzyme is a serine protease, a proteolytic enzyme. This enzyme hydrolyzes the proteins of the infected cell and also the proteins of the pathogen which is present inside the infected cell. So the proteins of the infected cell and proteins of the pathogen are broken down and destroyed by granzyme. And granulysin activates apoptosis of the infected cell. So the infected cell is programmed to die. So the infected cell will die and the pathogen which is present inside the infected cell will also die. In this way, the combined action of perforin, granzyme, and granulysin results in destruction and death of the infected cell and also destruction of the pathogen which is present in the or which was present in the infected cell. The second type of T cells are helper T cells, and they have a different function. But there are certain common features between helper T cells and cytotoxic T cells. But these two types of cells also differ in certain respects. The similarities between helper T cells and cytotoxic T cells are both of them possess T cell receptors. The T cell receptors are made up of alpha chain and beta chain. The cytoplasmic tail of T cell receptors is associated with the same CD complex having items. Both the cells, helper T cells and cytotoxic T cells, possess co-receptors, and this co-receptor is associated with LCK. The cytoplasmic portion of co-receptor is associated with LCK. The differences between helper T cells and cytotoxic T cells are the co-receptor in cytotoxic T cells is CD8, but the co-receptor in helper T cells is CD4. And secondly, the receptor of the cytotoxic T cell recognizes a foreign peptide which is bound to MHC protein, whereas the T cell receptor of helper T cell recognizes a foreign peptide bound to MHC protein. The CD4, which is the co-receptor present in the helper T cells, is made up of a single polypeptide chain, and this chain has got four domains which are known as D1 domain, D2 domain, D3 domain, and D4 domain. So this is the CD4 protein having the four different domains. We have seen that the helper T cell, the receptor of the helper T cell recognizes foreign antigens only when they are combined with MHC2 proteins. And MHC2 proteins have a very restricted tissue distribution. They are present only on macrophages, follicular dendritic cells, and B lymphocytes. So these three types of cells which present antigens to helper T cells are known as antigen-presenting cells. So the antigen-presenting cells are macrophages, follicular dendritic cells, and B cells or B lymphocytes. And these cells present the antigens, the foreign antigens to helper T cells. So these are the three types of antigen-presenting cells. The T cell receptor of a helper T cell binds to foreign antigen or foreign protein which is combined with, which is bound to MHC2 protein of the antigen presenting cell. And then the co-receptor, that is CD4, binds to the MHC2 protein. The T1 domain of CD4 binds to beta 1 domain of MHC2 protein. And when this binding occurs, an intracellular tyrosine kinase, which is known as FYN, is activated by a protein known as CD45. So FYN, a tyrosine kinase, which is normally inactive, is activated when the T cell receptor and the co-receptor of a helper T cell bind to a self MHC2 protein plus a foreign peptide. The active FYN now phosphorylates the items of CD3 complex. The phosphorylated items are the docking site for ZEP70. So they attract ZEP70. ZEP70 is bound to the items of the CD3 complex. Now, as yet, ZEP70, which is a tyrosine kinase, is inactive. But now the LCK, which is associated with the cytoplasmic portion of CD4 and which has become active, this LCK, active LCK, phosphorylates ZEP70 and ZEP70 now becomes active, an active tyrosine kinase. And this active ZEP70 phosphorylates the tyrosine residues of some target proteins which are present in the cytoplasm of the helper T cell. And this results in the release of certain proteins from the helper T cells. And these proteins are known as cytokines. Or since they are released from lymphocytes, they are also known as lymphokines. So a number of cytokines or lymphokines are released from the helper T cell. So we have an infected antigen presenting cell, an antigen presenting cell which has been infected by a pathogen. Some of the proteins of the pathogen are broken down inside this cell and a peptide fragment is attached to MHC2 protein, and this peptide is displayed on the surface of the antigen presenting cell. This combination of a foreign peptide plus the self MHC2 protein will be recognized by the T cell receptor of some helper T cell which is complementary to the foreign peptide. So this is the T cell receptor of a particular helper T cell, which is complementary to this foreign peptide. So this T cell receptor binds to the foreign peptide as well as the self MHC2 protein. The co-receptor CD8 also binds to the self MHC2 protein. The cytoplasmic tail of CD4 is associated with LCK. The cytoplasmic tail of T cell receptor is associated with CD3 complex. CD3 complex is made up of epsilon, delta, this gamma, this epsilon, and these two zeta chains. These chains possess the items. These are the items present in the cytoplasmic portion of CD3 complex. And these items, when they are phosphorylated by F by N, attract ZEP70. ZEP70 is bound to these items, and ZEP70 is phosphorylated by LCK. So now this ZEP70 becomes active. It phosphorylates the tyrosine residues of some target proteins in the helper T cell, and the helper T cell now releases the cytokines or the lymphokines. The lymphokines which are released by the helper T cell include various interleukins. Interleukins are abbreviated as ILs. So, a number of interleukins like IL-2, IL-3, IL-4, IL-5, etc. are released from the helper T cell. A protein known as granulocyte colony stimulating factor or GCSF is also released. Another protein, granulocyte macrophage colony stimulating factor or GM-CSF, is released from the helper T cell. A protein known as tumor necrosis factor or TNF is released from the cell, and interferon gamma is also released from the helper T cell. So a number of lymphokines or cytokines are released from the helper T cell. These lymphokines will produce various effects. IL-2 has a kind of autocrine action. It is released by helper T cells and it acts on helper T cells. It stimulates the multiplication of helper T cells, so more helper T cells will be formed. IL-3 stimulates the proliferation of stem cells in the bone marrow, and stem cells are the precursors of B lymphocytes and T lymphocytes. IL-4 stimulates the proliferation of B lymphocytes, T lymphocytes, and mast cells. IL-5 stimulates the multiplication of eosinophils. IL-10 stimulates the multiplication of mast cells, and it also increases the synthesis of MHC2 proteins in B lymphocytes. GM-CSF stimulates the multiplication of granulocytes and macrophages. Interferon gamma activates the macrophages, and it also activates the natural killer cells or NK cells. Tumor necrosis factor also activates the macrophages, and some of the lymphokines attract phagocytic cells to the site of the infection. So, the release of lymphokines results in an increase in the number of helper T cells themselves, an increase in the number of cytotoxic T cells, an increase in the number of B lymphocytes, and from B lymphocytes, plasma cells will be formed. There is an increase in the number of antigen-presenting cells, increase in the number of phagocytic cells, and activation of phagocytic cells. In this way, the release of various lymphokines from the helper T cells results in activation of innate immunity as well as adaptive immunity. In adaptive immunity, humoral immunity is activated and cell-mediated immunity is activated. So both innate and adaptive immunity are geared up to fight against the pathogen. And a coordinated and effective immune response is mounted against the pathogen. The helper T cells are known as helper cells because they respond to calls for help. This call is given by the antigen presenting cells which have become infected, and the call is in the form of a foreign peptide attached to the self MHC protein displayed on the surface of the antigen presenting cell. So when the antigen presenting cells which have become infected display a foreign peptide on their surface, it is a call for help, and the helper T cells respond to this call for help. The helper T cells cannot destroy the pathogen or the antigen. They have no direct role in destroying the pathogen or the antigen. They activate different arms of the defense system. Innate immunity is activated, humoral immunity is activated, cell-mediated immunity is activated, and the helper T cells coordinate the actions of innate immunity and adaptive immunity to mount an effective immune response against the pathogen. And therefore, the helper T cells are described as the commanding officer of the defense force. If helper T cells are depleted or debilitated or destroyed, the entire immune system will be crippled. The third type of T cells are suppressor T cells. They are also known as regulatory T cells. Like other T cells, they possess the T cell receptor. And the co-receptor which is present in these cells is CD8. The function of suppressor T cells is to regulate the immune response. They regulate the magnitude of the immune response, and they shut down the immune response when the pathogen, the infecting microorganism, has been destroyed. The suppressor T cells also release some proteins which are known as lymphokines. But these lymphokines are released when the immune response has achieved its goal. The goal is to eliminate the pathogen. And when the pathogen has been eliminated, the suppressor T cells release their lymphokines. And these lymphokines send signals to different arms of the immune system to seize their attack, to stop their attack. Moreover, the suppressor T cells have some role in preventing an immune response against self molecules. Some of our own molecules possess all the properties of antigens, but they are not foreign chemicals. And our immune system does not act against these potential self-antigens. And suppressor T cells have some role in preventing an immune response against the self molecules or the potential self-antigens. The fourth type of T cells are memory T cells. These cells retain the memory of the antigen after the first exposure. So when T lymphocytes encounter a pathogen for the first time, some of the T lymphocytes are converted into memory T cells. These cells retain the memory of the antigen. The memory T cells spread throughout the lymphoid tissue. And if the same pathogen or the same antigen enters our body again, the memory T cells are rapidly released into circulation from the lymphoid tissue, and this helps in mounting a quick immune response against the pathogen or the antigen. Our immune system is made up of different arms like innate immunity, humoral immunity, and cell-mediated immunity. But these arms do not act in isolation. They act in coordination. They coordinate with each other. Innate immunity acts in coordination with adaptive immunity. In adaptive immunity, the actions of humoral immunity and cell-mediated immunity are coordinated. So because of this coordination between different arms of the immune system, we are able to mount an effective and integrated immune response when we are challenged by any pathogen. Thank you.